So under the Classworks tab in your Google Classroom, for virtual students, since you're a virtual student, you will identify the Microevolution lab. And on the Microevolution lab, you will want to open up your document. So you may need to click View Document in order to see the Google Doc. Some people have had to click the View Document in order to see it. Some people have not. So some people, their assignment showed up over here. Okay, so you'll click on the document. And your document has your name in the title because a document was made specifically for you to record your answers. You'll want to open up the usernames and passwords file. And you'll need to go to your class period in the file and identify your username and password for the Gizmos. So you'll just scroll until you find your class period and your username and password. We're not submitting any of our work on Gizmos, and the Gizmos resets when you log out of it. So there's not a big deal that it's not a big deal in this situation that somebody could see your username and password for the Gizmos in particular. These are temporary accounts. They will expire relatively soon. And all we're doing is accessing the virtual lab. We're not submitting any of our work on there. So you just find your username and password. And I promise you, these were randomly generated. There's no meaning behind the usernames and passwords. So now that you have your username and password, you will click on the Explore Learning Gizmos link. And you will click Log In, and you'll type in your username and password. So that username and password that you just determined on Google Classroom. And when you log in, it will say your class period at the top. But other than that, it should look like this. You should see the Microevolution Lab, and you'll click Launch. I understand right now you have my shared screen open, so this would be challenging. But when you're working independently to finish this virtual lab, I strongly advise you to try and have two windows open simultaneously so that you can have your document right next to your gizmos, because it makes it just a lot easier to be able to see them both at the same time. Okay, so we'll start with a little bit of review. So we've talked a little bit about sickle cell anemia. And just as a reminder, many people from warm regions carry an allele that provides resistance to malaria. However, 2 copies of this allele causes a deadly blood disorder called sickle cell anemia. Now, when it says deadly blood disorder, it doesn't mean that sickle cell anemia kills everyone who has it, but it can be deadly. Remember that red blood cells should be disc-shaped, but with sickle cell anemia, they are shaped like a sickle, at least some of them are, and those sickle-shaped red blood cells can clump up, and that can be painful, can also be dangerous. They can form blood clots, and blood clots can cause strokes and even death. But the thing is that the little r allele does provide resistance to malaria. Remember, malaria is an illness that is transmitted by mosquitoes and is common in warm regions. There is medication now to treat malaria, but it was developed in the last 100 years. So for centuries, there was no treatment for malaria. Many people died. People still die from malaria to this day, even though there is a medication that can be used to treat it. Not everybody has access to it. So having one little r, having one of the sickle cell alleles, was actually very beneficial for a person, because the big R allele is the normal shaped red blood cell, and that's dominant. So that means that a person who is big R little r gets to have a normal shaped red blood cell, but they still get to have resistance to malaria. So they have the best of both worlds. They have a normal shaped red blood cell while also being resistant to malaria, meaning they're more likely to survive than their peers, because their peers were dying from malaria at high rates. But these people that have one big R and one little r allele were able to survive the malaria. The resistance to malaria doesn't mean that they didn't get malaria, but they were much less likely to die from it. They were much less likely to have a severe case of it. Now, just a reminder, what is an allele? An allele is a form of a gene. So remember that as humans, we have 23 pairs of chromosomes. You get one chromosome from each pair from each of your biological parents. So for the vast majority of the genes that you have two copies of them, two alleles, an allele being a form of that gene. The only exception would be biological males who only have one copy of the genes that are located only on the X chromosome and not located on the Y chromosome. But for all the other genes, you would have two copies of that gene because you inherited one allele from your biological mother and one allele from your biological father. So this question asks us, suppose two parents are healthy carriers of the sickle cell allele. That means that the genotype of each parent is big R, little r. How likely is it for a child of these parents to have sickle cell anemia, meaning being little r, little r? So if we look at this Punnett square, we see that of the potential offspring for these two healthy carrier parents, these two heterozygous parents, 1 out of the 4 options is big R, big R. So these offspring would have normal shaped red blood cells and no resistance to malaria. 2 out of 4 options would have big R, little r. So they would have normal shaped red blood cells and they would have resistance to malaria. And then little r is 1 out of the 4 options, 1 out of the 4 possibilities. And these individuals would have sickle cell anemia. So 1 out of 4, you're going to put the percentage in the box. Now, this text box is a little picky. You have to click in the bottom middle in order to be able to type. So you'll put the percentage. 1 out of 4 is what percentage? What probability? And then for the next question, do you think the allele for sickle cell anemia would be common in regions where malaria did not exist? So let's think about this. Mutations often happen randomly. But it's the ones that are beneficial that tend to be passed on from generation to generation. I'm not saying that negative ones are never passed on, but the ones that are beneficial are the ones that are more likely to be passed on because those are the individuals who are better able to survive long enough to have their own children and pass on their genes. Well, in areas where you have high rates of malaria, being big R, little r is ideal because these people with big R, little r have normal shaped red blood cells, but they have resistance to malaria. So they're not dying of malaria when their peers are dying of malaria. So they're going to live longer, are more likely to live longer and have their own children and pass on their genes. However, if you have a little r in your population, in your gene pool, then you always run the risk of somebody being born with little r, which would cause them to be classified as having sickle cell anemia and to have those sickle-shaped red blood cells. If there was no malaria in the area, there's no benefit to having a little r. If there's no malaria in the area, being big R, little r is not an advantage to an individual. It wouldn't make that individual any more likely to live longer, any more likely to outlive their peers because their peers aren't dying from malaria if there's no malaria in the area where they live. And you run the risk of somebody being born with sickle cell anemia. So especially before there were modern treatments, a person with sickle cell anemia, you would expect to have a shorter life expectancy because of the health complications. So they would be less likely to live long enough to have children. Again, this is specifically before modern health treatments. So even if this mutation happened randomly in an area where there was no malaria, there'd be no benefit to it. So it wouldn't be likely that it would be passed on long term. It would likely die out in the population or at least become very, very rare in the population because there wouldn't be a benefit to having that mutation. Okay. At some point, you'll need to watch this video that reviews the peppered moths. We've talked about the peppered moths and how the population shifted in color over time when the environment changed. So you'll watch that video and you will explain the changes that happened. Now we're going to take some time to go over the gizmos itself. So for the warm-up, we'll start here. If natural selection does not favor one trait over another, the frequencies of various alleles in a population will tend to be stable over long periods of time. So what does that mean? Well, you can have alleles, so alleles being different forms of the same gene. You can have alleles that have neutral differences. They're different, but there's not an advantage or a disadvantage of one compared to the other. So brown eyes versus blue eyes, that would be a good example here, okay? Those are two alleles for eye color. So they're obviously different forms of that eye color gene, but there's no advantage for survival of having brown eyes compared to blue eyes or having blue eyes compared to brown eyes. So the frequency of brown eyes and blue eyes remains relatively stable over time. Sure, there may be more of one than the other, but that frequency remains pretty consistent. It doesn't go up or down significantly. It stays pretty much where it's at. But what happens when one allele confers an advantage or disadvantage to an individual? These scenarios and others can be explored with the microevolution gizmo. So the feather color of the parrots in the gizmo is controlled by two alleles, big D and little d. So big D meaning a dominant allele and little d meaning a recessive allele. The big D allele is incompletely dominant over little d. What is the feather color of each parrot genotype? So remember, genotype is what's in your genes, what's in your genetic code. That's your two alleles. So the genotypes are listed here, big D, little d, and little d. And we're asked to give the feather color, so the phenotype, what we see. Now, up until this point, we've only talked about complete dominance. This is a situation with incomplete dominance. So let me point out the difference here. Brown eyes versus blue eyes are an example of complete dominance. Complete dominance means that if you have a heterozygous genotype, you will see the dominant phenotype and only the dominant phenotype. So just by looking at a person, you cannot tell whether they are big B, little b, or whether they are big B, because big B eyes will look exactly the same as big B, little b eyes. For example, I know my biological mother has blue eyes, so I know that I have to be big B, little b. But I have very dark brown eyes. Just looking at my eyes, you have no way of knowing that I have a little b allele. I only know that because I know that my biological mother has blue eyes, and a person who has blue eyes has to be little b. That's the only way that a person can have blue eyes, is to be little b. So in this situation, we have, of the offspring, we have three potential genotypes, big B, little b, and little b. But we only have two potential phenotypes. There's either brown eyes or blue eyes. Incomplete dominance means that the heterozygous genotype will have an intermediate phenotype, which is a lot of words to say that the heterozygous genotype is going to look like an average of the homozygous dominant genotype and the homozygous recessive genotype. We see this a lot in flowers. So you'll have a red flower and a white flower, and then the heterozygous will be pink. So in an incomplete dominance situation, we would have three phenotypes, three separate phenotypes, the red, the white, and the pink, instead of just two phenotypes. So if the red were completely dominant over the white, then these would also be red. But since the red is incompletely dominant, these heterozygous flowers are pink. So these parrots represent incomplete dominance. So the phenotype is what we see. So we need to identify what we see. So for the big D parrots, we see dark green feathers. For the little d, the homozygous recessive, we see yellow feathers. For the big D, little d parrots, we see light green feathers, which makes sense because the heterozygous genotype has an intermediate phenotype. So it's like we averaged out that dark green and the yellow, and we got light green. The fitness of parrots is determined by how well they blend into the background trees. So when an animal blends into its surroundings, we call that camouflage. So we're trying to figure out the camouflage of the various parrots here. In this case, we're going to set the fitness of the big D slider to 100%, and we're going to set the fitness of the other sliders to 60%. We're going to click Show Populations. Okay, and before we do anything else, the birds with which genotype blend into the background. So which genotype? Big D, little d, or little d. You got to figure out which ones are blending in best to their background at the moment. So you'll put that in the box here. Now we're going to click Begin. Let them live their little birdie lives. And then we're going to bring the predators in. So the hawks are going to get their food. And your table will not look identical to mine, but it will look similar if you've set yours up the same way. And so now you're going to answer which parents were killed by predators and why. So think about which ones blended in the best, which ones had the worst camouflage, and which ones were killed by the predators, and how does that relate to which ones had good camouflage and which ones had poor camouflage. Okay, there are three activities in this virtual lab: activity A, activity B, and activity C. You have to finish collecting all of the data for a given activity in a single session, in a single Gizmos login, because Gizmos does not save your work. So if you log out in the middle of an activity, you will have to restart that activity. So make sure that you obtain all of your data for a given activity in a single session on Gizmos. Now, you can do a session for activity A, a session for activity B, and a session for activity C, or you can do them all in one session, or you can do A and B in one session and C in a separate session, or other combinations like that. But you just can't log out in the middle of an activity, or you'll have to restart that activity. Okay, so for activity A, it says to click Reset. So we're going to click Reset. And it says to set the big D, and little d sliders to 34%. We're going to set to 34%. So make sure both of these are 34%. A deleterious allele is one that significantly lowers the fitness of an individual. So deleterious means that it's bad. It's bad for you. So the sickle cell allele for individuals with sickle cell anemia, it is a negative. Okay, that one is complicated in the sense that having one copy of it is beneficial, but having two copies of it is definitely negative. Now, with sickle cell anemia, that's a recessive allele, so you have to have two copies of the little r allele to end up with sickle cell anemia. Whereas activity A, we're going to model a deleterious dominant allele. So if the deleterious allele, meaning the allele that's bad for you, is a dominant allele, then both homozygous big D genotype and heterozygous big D genotype individuals will be at a disadvantage. Because if it's dominant, then you only need one of them for it to be a bad thing for you. How will allele frequencies change if a dominant allele is deleterious? So to model the effects of a deleterious dominant allele, we're going to set the fitness of big D, and the fitness of big D to 60%. We're going to set the fitness of little d to 100%. So based on the color of the trees, which parrots will be easiest for the predators to spot and kill? So you'll put your observation in that box there. Now you're going to predict how do you expect the proportions of big D and little d alleles to change in 5 generations, so over the course of 5 generations. So there's a few things that I want to point out. Right now, we have 170 individuals that are big D. Your genotype represents 2 alleles because you inherit one allele from your biological mother and one allele from your biological father. So that means that there's 170 plus 170 big D alleles in the gene pool at the moment because these individuals have 2 big D alleles and there's 170 of these individuals. And then there's 160 big D little d individuals. So that's another 160 big D alleles in the population. So that's 500 big D alleles total in the population right now, initially. For the little d alleles, we've got 170 individuals that are little d. So that's 170 plus 170 little d alleles, since these individuals have 2 of those alleles. And then another 160 little d alleles because the heterozygous individuals have 1 little d allele. So there's 500 little d alleles in the population right now. So right now, there's an equal number of big D alleles and little d alleles in the population. Remember, dominant does not mean more common. A dominant allele may be more common, or it may be less common. It could be very rare. Dominant just means that it asserts itself over the recessive phenotype. So it may be an incomplete assertion, like you have here with the pink, or it may be a complete assertion where you have here with the brown eyes over blue eyes. So big B, little b being completely brown eyes. But regardless, it asserts itself over the recessive allele. So to make your prediction about how the proportions will change, so right now it's a 50-50 split. There's an equal number of big D alleles in the population as there are little d alleles in the population. Each person or each individual, in this case we're talking about parrots, each individual has 2 alleles because your genotype is 2 alleles. So you need to take into account this situation and think about which parrots are more likely to live longer, and as a result of living longer, have more opportunities to have babies and pass on their genes. And then which individuals are more likely to be killed by the predators at an early age, meaning they won't be able to pass on their genes. And so there would be fewer of those genes passed on to the next generation. So which ones do you think will be able to have more babies? And as a result of that, what do you think will happen? Do you think that the big D and little d alleles will stay equal? Or do you think that you'll end up with more of one compared to the other one? And if so, which one? Okay, so we're going to click begin. Then we're going to come over here. So they're living their little birdie lives. We're going to bring a predator in. So we had some hawks come in. Now we're going to take this table and we need to put it into our Google Doc. Now, if you have 2 windows open side by side, it's pretty easy to just type these numbers in. But again, I know that right now you're looking at my shared screen, so that would be a challenge to do that. So another option is if you have a phone or a tablet, you could take a picture of this chart right here and then look at that picture to fill in your table on your Google Doc. Once you have your table filled in, we need to calculate the percentage of survivors for each genotype. To do that, we're going to divide the ending population by the starting population and then multiply our result by 100 to convert to a percentage. Now, your numbers are going to be slightly different than mine, so you want to use your numbers. For me, my ending population for the big D genotype was 108. And I need to divide that by my starting population, which was 170. And then I need to multiply that by 100. Now, this doesn't tell you what you need to round to, so I would suggest 1 or 2 decimal places, just round appropriately. So if I do 1 decimal place, the next digit is a 2, so that 5 stays a 5. For my big D, little d, my ending population is 90. My starting population is 160. And then I need to multiply that by 100 to get my survival percentage. I included both decimal places because this ended, so it made sense to just include the whole number there. And then all of my little d parrots survived. You may have had 1 or 2 that were killed by the hawks. If you had more than 2, double check and make sure that your numbers are appropriate here, that you set this the way that it told you to set it. You may have had 1 or 2 that were killed by the hawks, though, because camouflage is very useful, but it is not a guarantee of survival. So I had 170 survive. I started out with 170 little d, and then multiply that by 100, and I get 100% survival rate for my little d phenotype. So given the current time, I'm going to go ahead and move on. You will need to come back and analyze how the percentages of survivors relate to the fitness of each parrot genotype. So you'll want to look at the fitness levels, and you'll want to discuss which ones were more camouflage, which ones were less camouflage, and how that related to your survival percentages here. Now, I will tell you that these numbers, you're looking to have something in the 50s or 60s. If your percentages here are not in the 50s or 60s, double check and make sure that all of your information here, that you set your parameters correctly. And then this number should be 95 or bigger. If it's not, double check and make sure that you set everything appropriately. So you'll include your discussion there. Make sure you use full sentences that are grammatically correct. Okay, so we're going to quickly try and finish up activity A here so that you know what to do for the rest of the activities. So we need to simulate those 5 generations that we made a prediction about. So we're going to click breed. So they found a parrot partner. They laid some eggs. Now they're going to hatch the eggs. This chart here, you don't have to record anything from this chart. This is just letting you know what pairs you ended up with. So for me personally in the simulation, I had 15 dark green pairs, 30 dark green, light green pairs, 48 dark green, yellow pairs, 13 light green pairs, 34 light green, yellow pairs, and 44 yellow pairs. As soon as the next button appears, so as soon as it's not grayed out, you can click it. So they live in their little birdie lives. The predators come in. So see, in this generation, I had a yellow one that was eaten by the hawk. That happens sometimes. Camouflage is not a guarantee of survival. Now, you'll see that you have some other tabs over here. We could record the information in this table from here, but we'd have to do it separately on 5 different generations. So it's easier to just come over to this table, click on this tab, and use the information here. You can go through your full 5 generations and then record your table. So you'll breed, hatch, continue, the predators come in, breed, hatch, continue, the predators come in, and you'll do that to get to 5 generations. Now, if you accidentally click the button in extra time and you end up with a 6th generation, it's not a big deal. You're just going to record through the 5th generation in this table in your Google document. But again, it's not a big deal if you end up with an extra generation in your table here. You don't have to start over because you had an extra generation. You just want to record what you have from generation 0 through generation 5. And again, if you have 2 windows open simultaneously, it's pretty easy to just look and type in that information. But if that's a challenge, so if you've got a small screen or if you're also trying to look at my shared screen, then alternatively, taking a picture of it on a secondary device, like a camera or a phone or a tablet, and then looking at that device to be able to record your numbers would be very helpful. So you'll take a look at this table, and in full sentences that are grammatically correct, you will analyze the patterns that you see in your data. So which ones are going up? Which ones are going down? Are they going up quickly? Are they going up slowly? Are they going down quickly? Are they going down slowly? Are they staying steady? Explain all of those patterns that you see. Or which patterns do you actually see? Do they go up and then down? You know, something like that. Okay, so before we can leave, we need to obtain our graphs. At the very least, we need to obtain our graphs. For 8 and 9, you will need to include a written explanation about what the graph shows. You'll need to put that in your own words. But you should also include a picture of the graph itself. So if you click on the allele graph, and you want to make sure that you have your 5 generations completed, then you'll see a little camera here, and you'll click on the then your graph will show up. Now, to copy your graph or to download it, you need to right-click. If you have a Windows machine, or if you have a MacBook, or if you have an external mouse plugged in, then you probably have a physical right-click button. But if you have a Chromebook, especially if you've not changed your settings, you probably don't have a right-click button or even a right-click area on the trackpad. The default for a Chromebook, right-click, is a 2-finger tap. Not a double tap, a 2-finger tap. So you take 2 fingers and you tap the trackpad at the same time. So I use my pointer finger and my middle finger, and I tap the trackpad at the same time. That will bring up the right-click menu. You can then select Copy Image, and then go over to your Google Doc and do your right-click again, so that 2-finger tap on the trackpad. That'll bring up your right-click menu, and you can paste. Alternately, you could do Control-V there to paste. And then you'll want to resize it so that it's not huge. Now, you will need to come back and explain in your own words what you're seeing in this graph. But go ahead and put the graph in right now. In your genotype graph, you'll do the same thing. So you'll click on the genotype graph tab. You'll click camera. The graph will come up. You will 2-finger tap on the picture to get your right-click menu. Copy your image. Come over to your Google Doc. 2-finger tap. Paste your image. And then resize it. Now, at this point, you have all of your data for activity A from the gizmos. You haven't finished activity A in the sense that you still have explanations that you need to complete, but there's nothing else that you need to do in the gizmos for activity A. There's nothing else that you need to do on this website for activity A. So if you were to log out of Gizmos right now, you would not have to restart activity A because you have all the information that you need for activity A. So you need to make sure that you have your table complete, or that you've taken a picture of your table so that you can complete it at a later time, and that you have your pictures of your 2 graphs before you log out of the gizmos. Now, again, you can stay logged into the gizmos and finish your other activities, but I was just letting you know the minimum of what you have to have. So remember to do activity B and activity C in a single setting on Gizmos, because if you log out, it will erase all your data. So let's say you were on generation 3 in your table. Well, you'd have to start all over, because when you log back in, it's going to reset it and your numbers will not be exactly the same numbers. So you'll need to restart that activity if you log out during an activity. But at this point, I could log out and I would still be ready to start activity B in the gizmos. I've got to finish my explanations for activity A, but in the gizmos, I don't need anything else for activity A.